We are now ready to begin the last section of Chapter 13, which talks about gene regulation. Well, if we think about it, every cell has the same DNA. Every cell in your body has the same DNA, the same genes. But not every cell does the exact same thing. So there's something going on in your cells that say, oh, you need to be turned on and you need to be turned off. You're going to be a bone cell. You're going to be a muscle cell. And that's what we try to figure out. So the word that we use in biology is we say that we are regulating the expression of the genes. By the expression, we're saying what traits are allowed to be on and appear. So not to write, just to think about. There's a couple different dimensions of gene expression. So one of the dimensions is time. And if you look at these pictures, as this child grows from just being a 10-week-old embryo to a 14 weeks to a day old, all the way up to 18 months, there's changes that have taken place. And there's genes that have been turning on and turning off to allow these changes to happen. So time is one dimension. Again, still not to write, just to think. Space is another dimension. This very colorful picture is a fly embryo, and it's been color-coded to show the areas of the different genes. So different colors are going to develop into different parts of this fly. So one end will be a head, and another color will be a wing, and another color will be an antenna or something along those lines. So, space, where the genes are located in the actual organism, will determine the expression of the genes. And lastly, still not to write, just to think about the abundance. This picture here is color-coded to show how much of a gene is present. So the darker blue is showing a lot of this gene being present, and then the lighter blue is showing it not being as present. So these will affect the function, whether maybe it's going to become a heart muscle or a kidney muscle, will be determined by how much of that gene appears. So if we look at this cute little cat, we can just see some of the reasons why genes need to be regulated. So just a very common cat, that pattern, that striped pattern, is gene regulation. Having chromosomes active or inactive is determining the colors. How this cat behaves is determined by genes. Tortoiseshell cats, animals in general, tend to be much more aggressive for some reason. So calico cats, if you own one, maybe it's a little bit naughtier than a cat that is a solid color. The metabolism, how quickly or slowly this cat is able to break down the food that it eats and use it for energy, is regulated by our genes. What made this cat a house cat and not a tiger is regulated by genes. And then this very sad picture of a mutation. We're seeing this poor little paw. He's got this weird thumb sticking out to the side. The cat doesn't know any better, but that's definitely some sort of a mutation. So how does this happen first in prokaryotes? Well, we have studied a not very exciting bacteria, the E. coli bacteria, found in our body, found in food, and it's one that makes people sick, very common. So we know lots about E. coli. And we've also watched E. coli that when it finds food, it knows, much like we do, that it has to make enzymes to break down this food. So it has to turn on the genes that will break down this food. But these genes don't need to be on all the time. So there's something going on in this very tiny one-celled organism that are causing the genes to be expressed. So we need to learn a couple new words, and Here's a picture of the E. coli bacteria down at the bottom of the page. The new word that we need to know is the word operon. And what an operon is it takes that promoter and a gene and they combine together to cause something to happen. And what it does in E. coli is this operon causes the gene to get switched on that says go to work. And so in the presence of a particular food, in this case we're going to use lactose, a sugar that's found in milk, this bacteria, when lactose is present, this bacteria will be able to digest it. And this operon that does turn on this gene is called the LAC, lac, for lactose, operon. Now, it doesn't need to be on all the time, so the genes get turned off by repressors. And we will talk about that on later slides. And it only begins to work when lactose is present. So it's not constantly trying to break down lactose, only when lactose is present. So as we take a look at this small piece of a gene, we can see at the beginning we have our promoter. And if the promoter is present, RNA polymerase comes in, connects to the gene, and we start transcribing. So we're going to start moving along the gene in this direction. Now, next to that, we have our operator. And we can just think of that as the O region versus the promoter, which is the. That operator acts like the on-off switch. So you can sort of see with this little loop, are we going to let the RNA continue, or are we going to hold it right there and not let transcription occur? So if the repressor, the LAC, the lac repressor, is present, then we can't get through. Transcription is not going to occur. So we can see that it's definitely blocking the way, and our gene to break down lactose is turned off. Now we're going to add some lactose, so some sugar has been added, and what happens is that sugar comes in and it binds to the repressor protein. So you can see it's going to fit right into this spot. When that happens and this sugar has hooked up with the repressor protein, that repressor protein moves away, and now you can see that our transcription can occur. Our RNA can move down the gene and transcription can take place. And now our gene is on. It's activated and we're able to continue transcription and break down this lactose that is now in the body. So when that lactose is gone and there's no sugar left, the repressor will go back to its spot, blocking that promoter. Transcription can occur, and the gene is now turned off. That was in prokaryotes, a very simple one-celled organism. In eukaryotes, in us, more complicated. So we don't usually have operons in our body. Our traits, our gene regulation, are controlled individually. And it's a very complex process, much more complex than just whether or not this lactose can bind to this repressor or not. And the reason that it's so much more complex is that if you remember, our genes get edited. And we have introns and exons, parts that leave the nucleus and parts that don't leave the nucleus. And that complicates the procedure. So what a lot of our genes have is what's called this TATA box. A TATA box is just simply a region of the gene that has a series of adenine and thymine bases. And it could be any number of them, but it's only going to be the A's and the T's. So you can see here we have our promoters, we have our TATA box, we have our introns and exons. And what this TATA box's job is to do, we think, is that it's going to tell the RNA polymerase, hey, come here. This is the spot that you need to start at to do the process that we need to have take place. So before the box, we have the little tiny sequences that are our promoter genes, then our TATA box, then our introns and exons. And those enhancer sequences are going to be different for the different parts of the gene. And they are set up so that it's not a single protein that can allow this gene to be switched on. There's different proteins that can come in and hook to these different enhancer sequences. And basically what these do, these proteins that come in, can make transcription occur in a couple ways. And here's 3 ways. One is they can allow the chromatin to unroll, to open up, to expand, so that DNA translation and transcription can take place. Sometimes these proteins will act as kind of lighthouses for the RNA polymerase, and they'll basically be saying, hey, come bind here. This is the spot where you need to start. Or they can do the exact opposite, and they can block access to the genes and say, no, you cannot come in. Transcription is not going to take place. Translation is not going to take place. And we're going to inhibit this gene and not allow it to be switched on. So all of the cells in our body are basically the same, except sometimes the genes get switched on and sometimes they get switched off. How do they know what to turn into? So the process where they grow and develop into different types of cells is called differentiation. So the cells that make up your heart and the cells that make up your arms started out exactly the same, but they became differentiated. They became specialized, and the heart cells only know how to act like a heart cell, and your arm cells that make the tissue in your arm only know how to do that. And there is a special set of genes called the Hox genes that are in charge of this. Basically, they are the conductors. They tell every cell what you should be. What are you going to do? And what we are doing, we as the scientific world, is we are trying to control the expression of these genes. So by controlling whether these genes get switched on or off, we can change how a strawberry is formed. Or we can develop a certain type of tissue. Whether that's good or bad, that's, not for us to decide at this time, but it certainly is taking place. And these particular Hox genes are passed on through generation after generation after generation. So we can see a commonality if we look from the wolf to the present day dog. There's some common traits that are passed on, so they all have the same general characteristics. We obviously know it's a dog and not a bird or a person. This particular slide is just showing you an example of the Hox gene. So we can see, for example, this fruit fly starting right here. Says it's zipping all over. Here's a chromosome, and if you look down at the embryo, you can see that this part of the chromosome is responsible for making the end part of the fly. This yellow part is going in to make the thoracic region of the fly. This green part goes in and makes the head of the fly. If you look over at the mouse, you can see the kind of a similarity going on, that these genes are differentiated. And this little section here is responsible for making the end part of the mouse, which continues on into this portion right here. So the fact that we can control it leads into the question, should we control it? And if we should control how genes are expressed, should we control and create new subspecies, things like a labradoodle, which would have never happened in nature. We were forcing it to happen. And then if this is true, if we are going to manipulate these genes, who should be in charge? Who's going to police it? Who's going to say whether or not you can manipulate these genes? And then as we continue changing these genes and switching different parts of the gene off and on, who's to say that someone's not going to try to manipulate dangerous diseases? And we already have a concern about this, that we are looking at warfare where we're not using guns, but the ammunition would be diseases. And then finally, something to think about is, just because we make a new organism, how does that affect the organisms that already exist? We've seen problems with this already with species of corn. And as you make new species that perhaps have a dominant trait, what happens to those recessive traits that maybe were some really tasty sweet corn but are now being diminished because a more dominant species that's easier to grow and grows quicker is taking the place of those? So we really need to think about how will these new organisms change our environment. And then on the next few slides, I just have some pictures that show some of the things that we are doing right now. This slide shows a particular type of cow that has been genetically altered so that it would make more meat. Is this good? Is it bad? This is an Appaloosa horse. It does not develop in nature. We have absolutely created it for our benefit because it's beautiful, because it's unique. The wild mustard plant has been genetically altered and used to change the different vegetables that you see here, to make them grow better, to make them grow faster, to make them grow in a way that makes it easier to get them on the supermarket shelves, good or bad. It's kind of an interesting one. The outside looks like an orange, but the inside looks like a kiwi. Genetic manipulation. And then to end up, just a few slides that have questions. So if you could read through these and just jot down what you think the answers are in your notes. We will go over these in our next class. So which sequence shows the typical organization of a single gene site on a DNA strand? Number 2, a group of genes that operates together. Number 3, repressors function to do what? Number 4, which of the following is unique to the regulation of eukaryotic genes? And the last slide of this chapter, organs and tissues that develop in various parts of embryos are controlled by